Welcome to the second lecture on the retina. And so in this one, I'm going to be talking about photoreceptors and phototransduction. So in the previous lecture, we said that light hits the rods or the cone photoreceptors, and that light energy is converted to electrical signals. But how do they actually do this? How do they detect light? So that's phototransduction. It is the conversion of photons to electrical signals, which we mentioned last time are going to be graded potentials. And it does this with opsins. So here on the left, this is an opsin. It's a G protein coupled receptor that is covalently, it binds a chromophore, a molecule that is responsive to light. And this chromophore is 11-cis retinal. And so this molecule is responsive to light. When a photon hits it, it converts the molecule from 11-cis retinal to all trans retinal. And that conformational change activates the opsin that it's bound to. And so rods and cones are just packed with opsins. And that's based on their structure. So a rod, this big, long outer segment, where it gets its name rod from, it's just packed with a bunch of the membrane that's folded in on itself, and they're called discs. And within these discs are just a whole lot of rod opsins. And so rod opsins are called rhodopsin. We'll be mostly looking at rods when we're talking about phototransduction. Cones have their own opsins, and they actually have three different types: the S opsin, the M opsin, and the L opsin. The S stands for short wavelength, M for medium, and L for long, and they are best suited to absorbing light in the blue, green, and red range. But they're also packed. They're not quite as tall, but they still have a lot of stacks of their opsin. So this, having a whole lot of the molecule, allows them to be very good at capturing photons. So since I just mentioned the three different types of opsins in cones, it's worth talking about color vision. So each cone will, in the humans at least, each cone only contains one of these three opsins, either the short, the medium, or the long. But our brains cannot determine the color of a light source from a single cone. So if you just were recording from a single short wavelength cone, one of these blue guys, and you shone a light on it and it had a certain graded potential change, you wouldn't really know what color it was. Maybe it was a really bright light in this kind of bluish green range, and so it gave you a moderate response. Or maybe it was a really dim light in this blue range, this optimal range, and it gave you a medium response just because the light was dim, even though it was of the best color. So you really can't tell the modality, the color of the stimulus from a single type of opsin. You actually need to compare between different types. So if you get a response in both a green cone and a blue cone, and you compare the two, that enables you to start working out what color the stimulus really is. So that's how our color vision works, by comparing between these three different cones. So the more cones you have, the more fine comparison you can make. So that's why we're able to really well determine these colors in here between red and green, even though it's not a very big change in wavelength, but our red and our green cones have peak sensitivities that are just so similar. So for your first summary question, it's a little bit of a weird one, but I want you to answer: if you had the superpower to evolve a new opsin, a fourth opsin, where would you want its peak sensitivity to be for you to better be able to distinguish shades of red. So if you want to be able to distinguish red, do you want its peak to be shorter wavelengths than the short, between the short and the medium, between the medium and the long, or longer wavelengths than the long? So that brings us to color blindness. By far, the most common form of color blindness is red-green color blindness. And that's when you're missing or have a non-functional M or L opsin. So these are the two that are peaked very close together. And they're peaked close together because evolutionarily they diverged very recently. So they're very similar. They're actually copies of each other that had some small changes made so that they have these differences in peak responsiveness. And so if you're missing one of these, you're not color blind, really. You can still see color. You can compare whichever remaining one, if you have in the case of pronatopia, where you're missing your L cone, you can still compare between the S and the M cone. You still see color. You just are really bad at telling the difference between green and red because you can no longer make that comparison. And the same goes if you're missing M cone down here with deuteranopia. And so you've probably heard that red-green color blindness is much more common in males, and that's because these genes for both the M and the L cone are on the X chromosome, so they're more common in biological males. And there are other forms of color blindness, but they're very rare, like missing the S opsin would make you now no longer able to compare down in the blue range, but S opsin is not on the X chromosome, so it's actually very rare. It's only 0.0001% of the population. So let's get back to phototransduction. So we've said that you have light striking this 11-cis retinal, and then that molecule changes and activates its opsin. So here we're looking specifically in rod photoreceptors with rhodopsin. So now we have an activated opsin. What happens? And so I put this in text too because I found the picture a little bit confusing. But rhodopsin is a G-coupled protein, and when it's activated, it activates its G protein, and its G protein is transducin. So specifically, transducin goes from binding GDP to binding GTP. Now it's ready to do work. It dissociates off from the rhodopsin, combines with an enzyme called phosphodiesterase, and then this complex, so the transducin GTP plus the phosphodiesterase, it can then float over and start doing its enzymatic job, which is converting cyclic GMP to 5' GMP. So that's shown over here where it kind of is moved over to the membrane and is making the cyclic GMP into just GMP, 5' GMP. The cyclic GMP enables the opening of cyclic GMP gated channels. These channels that are normally, when there's cyclic GMP around, let in sodium and calcium. So this whole cascade causes you to have less cyclic GMP, which causes the channels to begin closing. And throughout this process, you get a lot of amplification. So each rhodopsin is able to make about 1,000 activated transducins. Each of these transducin phosphodiesterase complexes make about, or remove about 50 cyclic GMPs. And then every ion channel that's closed, you are not able to have about 10 to the 4th ions per second flow through them. So overall, the amplification you get from just a single photon striking one of the rhodopsin, you get about 10 to the 6th ions that are no longer flowing into the cell. So this massive amplification. And so this is what the response looks like in a rod photoreceptor to a flash of light. So you have a flash of light and they're doing various intensities of light, and we are looking at the membrane potential of the rod. And with a dim flash of light, you see a small hyperpolarization. And with a bright flash of light, you see these very strong and long, it goes for about a half a second hyperpolarization, or a little over half a second. And so these are the graded potentials we were talking about, where you can get a small response, a big response, or anything in between. But why a hyperpolarization? That's a little weird, and maybe you picked up on it from the previous slide where we were talking about closing a channel. So if you remember, in a dark state, when you're not activating these opsins, this cyclic GMP-gated channel is open, and calcium and sodium are just flowing into the cell. So they're positively charged ions flowing into the cell is going to depolarize the cell. It's going to make the cell more positive. At the same time, this wasn't in the previous slide, at the same time we have potassium channels. These are potassium leak channels that are just always leaking out potassium. So a positive ion leaving the cell is going to make the cell more negative, more hyperpolarized. And so these are both just open, and they're kind of counteracting each other. Positive comes in, positive goes out, and the cell ends up at some resting membrane potential. Then when you have light that activates the opsins, you get the whole pathway we just talked about, and then you get these channels closed. So now you no longer have positive coming in, but nothing has changed with these potassium channels. You still have positive potassium going out. So now the balance is shifted and you have a hyperpolarization. You just have positive charge leaving, but you don't have positive charge coming in. For your second summary question, I want you to answer, if you knocked out the gene for this potassium leak channel, how do you think it would affect the photoreceptors' ability to respond to light? So next I want to focus on the range over which your visual system has to work, and it's actually a massive range of light intensities. So at the lowest end of the range, at the absolute threshold down here, this x-axis is in candelas per meter squared, but this absolute threshold, if you measure it in how many photons are hitting the eye, you only need like 5 or 10 photons hitting your eye to detect that small amount of light. But then if you're out in the moonlight or indoor lighting or even sunlight, you're getting a lot more photons than 5 to 10. It's actually a log change of over 10 logs, so 10 to the 10 times more photons. So down here you're getting 5 photons, up here you're getting 5 times 10 to the 10 photons. And your visual system has to be able to detect these very dim lights, but also in the daylight be able to detect small changes. And this, to put this in perspective, what 10 to the 10 is, it would be like me asking you to build a kitchen scale that could both measure an apple, detect the weight of an apple, but also accurately measure the weight of the Willis Tower. It's just a mind-bogglingly large range. So how does the visual system handle this huge range without becoming saturated or not very sensitive? The first thing it does is it divides it up. So we already talked about rods and cones, and it divides up the work so that at these low levels, so scotopic light levels, that's low levels of light, only rods are active. They are more sensitive to light. Whereas in these middle light levels, around moonlight or so, rods and cones are both active. It's kind of the high end of the rods, the low end of the cones. And then in daylight level, only the cones are active. So it divides the labor so that when the rods are becoming saturated, the cones are beginning to be activated. But that's not enough to deal with the wide range of light intensities that we're presented with. So there's actually multiple stages of light adaptation in our visual processing. And we're just showing one of those examples that occurs in the photoreceptors, but there are others. So in the photoreceptors, calcium entry through the cyclic GMP gated channels actually inhibits the flow of more sodium and calcium into the cell. It inhibits the channel directly. It inhibits the turning off of activated phosphodiesterase, and it inhibits the turning off of rhodopsin. So with this, self-inhibition, we're able to get responses over a larger range of light levels. So we've already touched on some of the differences between rods and cones, and I'm just going to highlight those here. First off, as I said, rods have higher sensitivity to light. Cones have lower sensitivity. Rods converge more. So many different rods, 15 to 30 different rods, converge onto a single rod bipolar cell. Whereas cones, it's fewer, and in the fovea, it's actually a 1-to-1. One cone converges or links up with just one cone bipolar cell. And this also helps the rod system be more sensitive. You're having more sensory cells collect light information to pass on to the bipolar cell. So there's more sensitivity of the rod pathway. Rods are also slower in response. That's partly due to how they amplify more, giving them more sensitivity, but they kind of sample over a longer period of time. Whereas cones are faster in their kinetics. So you can see this very rapid inward current. Inward current is indicative of hyperpolarization, then outward current. And finally, we already touched on cones are involved in color vision because they have those three different kinds of opsins which they can compare. Rods only have one opsin, the rhodopsin, so it really can't tell color. It can't compare amongst, different wavelengths. And that's why when it's really dark and only your rods are active, you are colorblind. If you're walking around at night, you can't tell what the color of things are. So now let's talk about how these photoreceptors are distributed, and it's not even in the human retina. The fovea, which we already talked about, it's that area of very high visual acuity. It is just packed with photoreceptors, but it's only packed with cones. There's actually no rods in the fovea. Even though the human retina is actually 95% rod overall, there's just none of them in the fovea. So the periphery is very rod dominated. So here you can see so 0 being the fovea, the very center of the retina, a big spike in the number of cones with no rods. And then as you go out, you get many more rods and very few cones. So the fovea has very high acuity because of all those cones, but in the very center of the fovea, we have the most high acuity, the highest acuity, because we have all those cones packed in, and we actually exclude a lot of the rest of the retina. So right here in this avascular zone, which is the very center of the fovea, everything gets squished out to the side. So the cone photoreceptors and their outer segments are here, but then they start stretching out laterally. And so the bipolar cells and the ganglion cells are actually excluded from this region. And then also there aren't any blood vessels in the way here. And so this gives light, the most direct path to the outer segment of the cone and gives us the highest acuity vision in our whole retina. So for the final summary question, I want you to answer, If all of your cone photoreceptors died, but the rod photoreceptors remained healthy, what part of your visual field would be most damaged, would be most affected? Alternatively, what part of your visual field would still be fine for the most part? What parts of the world could you still see?